Hello and welcome to FameLab Ireland, the final 2021. My name is Jonathan McCrae. I'm a science broadcaster and communications trainer, and it is my great pleasure to be your compere this evening. This is the 9th and final FameLab. It's just me, isn't it? I can imagine you saying, it is a sad occasion in some respects. FameLab is the science communication competition and training program that was founded over 15 years ago by the Cheltenham Science Festival, and it's delivered by the Cultural Relations Organisation, the British Council. And I've been with them all of that time. And while it is sad to say goodbye, I think it's appropriate to kick off tonight by reflecting a bit on what we have achieved over those 9 years. And I got on board at the very beginning and have been delighted to be working with the British Council for such a long time. And it is a really unique competition because I've had the pleasure of working with young researchers who are enthusiastic about their science and want to talk about it to the world. And while not everyone is young in the traditional sense, they are young in the way that they are curious and have an enthusiasm for their subject, which is absolutely infectious. And I have worked delivering the masterclass and hosting this every year for 9 years. And I have to say, I've been humbled to be in the presence of such enthusiastic and brilliant minds. We've done a huge amount of work with partners over the past number of years. We've had 25 partners from various institutions and broadened our network. But the real thing for me is that over 1,000 professionals and students working or studying in STEM have taken part in FameLab over the years here in Ireland. And 100 of those FameLab finalist alumni still get together and work beyond this event that you see here tonight. They have done talks across the world and really spread their enthusiasm for science much further than just the FameLab community itself. It has to be said that this wouldn't have happened without the support of funders like Science Foundation Ireland, sponsors like Henkel and CPL, and the amazing team at British Council and their supporting partners who have provided the technology that allows us to do it tonight, but also delivered for us in the venues. So thank you so much to everyone who's taken part so far. And it has been a really pleasant journey for me personally. Right, enough reflection and introspection. It's time for an injection of perfection. Please give a warm reception for our collections of judges who will perform an inspection, dissection, rejection, and ultimately an election. They are Dr. Ruth Freeman, who is a Director of Science for Society at Science Foundation Ireland. She has a PhD in genetics at Trinity and has spoken at countless national and international science conferences. At SFI, she's a really important remit that's very relevant to us here tonight because she oversees shaping and funding programs and projects to do with science communication here in Ireland. She's also a regular contributor to Ireland's greatest radio show, Future Proof. Ruth, you're very welcome. Our second judge is Tomas Sirkovic. He was appointed CEO of Business in the Community Ireland 3 years ago now. He is a member of the board of CSR Europe since 2008 and the World Business Council of Sustainable Development. He also sits on a number of different boards, including Young Social Innovators. He's been on judging panels for a number of initiatives, so he's going to bring his expertise in this area, including SEI Sustainable Energy Awards, Limerick Chamber Regional Business Awards, the Google Impact Challenge. So he is going to know what to look out for as a judge. Tomas, you're very welcome.
Thanks, Jonathan.
And our final judge was listed in the Progress 1000 London's Most Influential People. Fran Scott is an accomplished engineering presenter, maker, and pyrotechnician. Fran was a series judge on Channel 4's Lego Masters 3 years ago and is best known for her playful and often fiery demonstrations on the CBBC. That's too many B's. CBBC show, Absolute Genius with Dick and Dom. A scientist by training and an engineer at heart, Fran manages to fuse humor and science to inform and entertain. And for Fran, science and entertainment is at the heart of what she does. Seems to have struck a chord with those wanting information as her shows have been so successful. Fran, we're delighted to have you with us. Thank you so much for joining us as our final judge.
Great to be here. Thank you.
All right. Time for the rules. Each contestant will give a 3-minute talk on science or technology, engineering, or maths. They're not allowed to use PowerPoint or add any effects to their videos, and they can use props, but only what they can fit on screen. After each contestant performs, the judging panel will have around 2 minutes to give some feedback, ask some questions, and probe to see whether or not our FameLabbers know their science. Contestants will be judged on content, clarity, and charisma. Content, they have to have their fact right. They have to know what they're doing. Clarity, is it clear? Do we understand the idea that's being explored? And charisma, have they got that X factor that we look for in the performers of FameLabbers? Let's meet our first contestant, and he is Jack Daly. Jack is a complex individual who straddles the line between genius and madman. His words, not mine. His research is as diverse as a Christmas selection box, with interests in plant biotechnology, gut microbes, and human physiology. Jack developed the skills of science communication to keep him off the mean streets, so he says, and he avidly uses these skills as a PhD candidate at UCC. This talk is about spitting, which hopefully requires no demonstration. Over to you. The best of luck as our first FameLabber of this evening, Jack Daly.
I'll be honest, the first time I saw the test for COVID-19 infections, I was afraid. As that swab was inserted through the nose right to the back of the nasal cavity, I was overcome by fear. Because what if I liked it? What if I was the one weirdo on the planet that actually enjoyed the test? Now the reality is that I hated the experience, like so many other people. So why do we use this test, and is there a better way? Well, first let's discuss the infection process. So you're a close contact of someone, and now the virus is in your body. Now the spike proteins of the virus are going to attach to a protein on your cells called ACE2, just the way a key fits into a lock. Now this ACE2 protein is actually found on cells throughout your body. So the virus invades the cell, hijacks it, makes millions of copies of itself, and then exits. From there it can infect other cells or other people. Now to stop the spread of disease, we have to know who's infected. We do that by sampling people and testing for the presence of the virus. Now, we can either use the antigen test that detects the proteins of the virus, or we can use the PCR test that detects the genetic material of the virus. The PCR test is much more sensitive, by the way, and therefore the gold standard. But where do we get a sample for the tests? Well, COVID's a respiratory disease that attacks the cells in your airways, like the ones at the back of your nasal cavity with ACE2. That's why we swab there. But remember, other cells in your body have ACE2 also. In fact, infected cells in your gut actually shed the virus through feces, and we can measure the spread of COVID by testing wastewater facilities. That's not the better method I'm promoting, by the way. Okay, so another hotspot of ACE2 cells is the lining of your mouth, which poses an even better method for testing, saliva or spit. Simply spit into a sterile tube and send it away for testing. So is this better than a nasal swab? But I think you all agree you're much more likely to spit into a tube than be nasally mined. Also, you can prepare these samples at home. No more queuing at expensive to run test centres where healthcare workers risk infections every day. It's so simple that we could test entire schools, workplaces and hospitals rapidly, even picking up people without symptoms. You simply spit into a tube at home and drop it off at a collection box, like a post box. A disgusting post box, filled to the brim with spit. But you can also use this test to test for antibodies towards COVID, showing whether you have immunity to the disease through either previous infection or vaccination. Now, all these benefits mean we might see widespread rollout of the saliva test very soon. In fact, the NBA tested many players and staff last year using this method. So you see, saving the world by stopping COVID is as simple as spitting into a tube. It's the wrong tube.
Oh, it's always good to start with a funny one. That was very funny, Jack. I laughed twice out loud. Very good. All right, let's go to our judges. Ruth Freeman, let's go to you because I know SFI they did the rapid response COVID program, so you know quite a bit about this. What are your questions for Jack, please?
So, Jack, if this is so much better, why are we still using PCR testing?
Well, the PCR testing is still underlying this. So it's the sampling method is saliva.
Okay.
So what I'm actually challenging here is the old nasal swab.
Okay.
So ultimately, the PCR test will still be the gold standard test. Now, the antigen test has been trialed with it, but it hasn't been that successful. But
Okay.
you have to remember that the antigen test has been designed with the nasal swab in mind. So it's not picking up things that might be interfering with saliva and the test. It's kind of like spitting onto a pregnancy test. It's not really going to work, So, yeah. So, I mean, ultimately, the PCR is still the underlying mechanism of testing.
Okay. Thank you.
This is more disgusting than I thought it would be, Jack. Well done. Tomas.
The challenge that I have for you, Jack, is what if I am sick and I ask my brother or sister to spit on my behalf and put the tube down the beautiful letterbox that you were describing earlier? How do we deal with that?
Well, that's a good question. It's ultimately the same idea as the people who perform the rapid antigen tests at home and people who are lying about this. So, I mean, ultimately, that is just going to be a public health measure that we have to address. And you have to realize, I mean, the only way to combat that is for people to realize the responsibility on themselves for the greater community. So I think, I know it's a problem with the spit, but it's already a problem that exists today with the antigen test. So I think it's just public health. That's the way forward.
Okay, enough talking about spit.
Fair enough.
Jack Daly, everyone, well done. Thank you very much. Thank you to our judges. Well done. That is our first FameLab talk over and done with. And on to our next. It is by Ifulo Tembi, or Tembi for short, Fashina. She moved to Ireland from Nigeria to study human genetics in Trinity College Dublin. Then she ventured out into industry, working on regulatory reporting for clinical trials. She's now a PhD student at the McCoy Lab in RCSI, brilliant lab, I know it well, where she uses bioinformatics tools to explore how microRNA variation influences things like MS. She is passionate about making science accessible for non-scientists and telling lesser told science stories. She enjoys reading fiction and doing aerial sports. So will she sail through to the international final, or will her parachute fail to open? It's time to take the leap and find out. Tembi Fashina's talk is called Goldilocks, Getting Dosing Just Right on the First Try. Tembi, over to you.
Do you remember Goldilocks? She's the porridge lover that snuck into the house of the three bears and went into their kitchen to taste their bowls of porridge. She tried the first bowl, found that was too hot, before she tried the second, found that was too cold, and then tried the third, which was just right. Well, we have something in common with Goldilocks, and that is that when we need to use medicines, sometimes doctors have to try different doses before they get it just right. And that is because genetic differences between people mean that our enzymes break down drugs into their active forms in slightly different ways. And so the standard dose does not always work. And we need drugs to work, especially when patients are in severe pain and are suffering. So one way around this is to test which versions of drug processing enzymes patients have, and then use that information to choose a dose on an entirely different drug. So that sounds simple, but can we do this for every disease and every drug? Well, it depends. If drug A is broken down by enzyme A, then we can see how changes to enzyme A affect drug A. But if drug A is broken down by multiple enzymes, it's much more difficult to keep track of how changes to these affect drug A. But luckily, there are some families of drug-enzyme interactions that we can keep track of. And one enzyme family that's involved particularly is called cytochrome P450. This has 57 different members. One of the most important is called CYP2D6. This particular enzyme breaks down up to 1/4 of all known drugs, and it also has up to 100 different versions that affect its drug processing abilities. And this is important when you think of its targets, like a painkiller called tramadol. Now, tramadol is broken down by CYP2D6, and some versions of CYP2D6 give too much tramadol, others give too little, and some give just the right amount. And in a large group of people, about 10% would have too little, while about 2% would have too much. And having too much tramadol can feel like experiencing an overdose with symptoms like confusion, nausea, and shallow breathing, while having too little means that patients are still suffering. So going from that group back to Goldilocks, this time she has a severe toothache, and so she's breaking in to steal painkillers instead of porridge. What drug and what dose should she use? Well, without genetic testing, this could feel like having to taste a room full of porridge. But luckily, we now have databases with drug versions and enzyme versions that work together well. So while we can't predict how patients will react to every single drug, for some, we can get the dose just right on the first try.
Very good, Tammy. I've always found it weird that we give the same dose of drugs to someone the size of Minnie Driver and someone the size of Shaq O'Neal. Like, adults dosing really doesn't make a huge amount of sense to me. Really interesting talk. Let's start with Fran, shall we? Fran, welcome.
Thank you. So with this, is this actually in process at the moment, or is this something that will come into the medical industry in years to come?
So this is a great question, because essentially the information is already out there and it depends on each country's regulatory processes. In the Netherlands, there's a working committee that essentially works towards using this for specific drugs. So they're using it for painkillers, they're using it for, I think, anticoagulants as well, and for HIV. And then there's a working committee in the US as well that's really trying to push this. And I think it's already approved for certain drugs. So absolutely applicable for certain conditions and certain drugs, but not like on a whole scale.
Brilliant. Thank you.
Ruth?
A bit like you, John, I was wondering, actually, you were talking about size, but are there gender differences that we can easily pull out as well using this kind of technology?
This is another fantastic question, but I think with the X chromosome and with studies that look at the genetic implications of like drug use and everything, it's a bit complicated because we have to have the numbers. And then when we're looking at genetic studies, when you're looking at the cases, the controls, you have to look at huge populations. You have to control for ethnicity. You have to control for other drugs that they're using. So far, there's not much on a whole, on a large scale, but I'm sure that there are some differences. You know,
and hormone cycles, women are so inconvenient.
But even speaking of hormones, like there's some medicines that you can't use with, say, tramadol because of serotonin and interaction and serotonin syndrome. So those are already in part and parcel of the whole idea of looking at which genes are working together. So it's a great question. I would love to have an answer for that in a few years.
Very good. Thank you very much for that, Tammy. Congratulations on your talk. Our third speaker is Mariana Silva. She's originally from Portugal, and she came to Ireland as part of her PhD program as a Maria Sklodowska-Curie Fellow. Her research focuses on developing a method to produce tiny particles of drug molecules that could get rid of the need to take daily pills. She says it will make them a thing of the past by turning them into a monthly injectable. Outside the lab, Mariana enjoys attempting photosynthesis while reading and is fascinated by outer space. So it's time to roll up our sleeves and take Mariana's medicine. Will the side effects include delirium for our judges or fatigue? Let's find out, shall we? Her talk is called Long-Acting Injectables for Long-Lasting Health. Please welcome Mariana Silva.
I'm a free woman. I can do whatever I want, whenever I want, or at least most of the times. Sadie is not lucky as I am. Sadie is from Bangladesh. She's the same age as me, but she has 2 kids and another one on the go. She's a stay-at-home mom. Every day to get food, water, and other supplies, she has to walk long distances, around 15 to 25 kilometers barefoot, carrying a child in her belly, another one at the back, another one at the front, and a pile of sticks on top of her head. So Sadie likes to take contraceptives. She cannot afford to have more children. The thing is that there are still a few issues with contraceptives. They don't last long enough, they may cause adverse effects, they are costly, and the doctor is really, really far away to get them. So, as Cristiano Ronaldo says, today I'm not here to tell you about my dream, but to show it. So, what are these long-acting injectables? So they are kind of like a vaccine. They are injected into the body. The thing is that instead of activating the army that is your immune system, they inject a drug that is slowly released. So you don't have to take pills every day for months. In industry, to produce these particles that will make these long-acting injectables, we start from bigger ones and then we mill until we get smaller particles. The thing is that these particles are flawed. And when they interact with the body, they may cause adverse effects. Also, the machinery used to produce them can cause metal contamination. And as you can imagine, there are loads of energy involved in the process, so the final product is costly. What I'm trying to do to solve this problem is to create kind of like a farm, where I start to grow the particles from the molecular level until they reach the size that I want. Then I disperse them in a media that the body likes, and then ready to inject. But what is so special about these particles? Their crystalline structure is so defined and is so stable that it will cause the drug to be slowly released, so you have that long-acting effect. Okay, we solved that problem. But then we still have the problem of how do we make it more accessible to Sadie so that she doesn't have to walk long distances to get it? Microneedle arrays. I bet that everyone has seen the studs on the football boots that kids use to play soccer, football, or rugby. So this would be like a tinier version of this that would fit the top part of your finger, would fill the drug solution in the tiny needles, and then just by pressing it against your skin, the drug is injected into your body, as simple as that. So when all of this becomes a reality and we are close to make this happening, Sadie and her family will be healthier. Sadie will be able to work under no wage, and their kids will be able to go to school. This is how you get countries out of poverty. So let me ask you a final question. Why does it sometimes take so long to make these problems and dreams a reality? And why does it sometimes happen so fast? Thanks.
Really good, Mariana, and certainly something to contemplate, particularly considering the inequality of healthcare we've seen over the past year. Let's hear from Tomas. Do you have any questions for Mariana?
Yes, thanks, Jonathan. And I'm thinking about Sadie and her daily journey. So, Mariana, how do you think we could scale that up in terms of large volumes of manufacturing? Because that's quite a transformation in terms of the manufacturing of the drugs.
Okay, so these long-acting injectables already exist. And the thing is that I'm trying to scaling the bottom-up process. So instead of milling them, that's actually the current gold standard procedure in industry. We have to use a bottom-up production that obviously will require some adjustments, but that's what I'm trying to do to make it possible. And the thing is that it's like the social part and the healthcare system is a problem that these countries don't have. But still, there are foundations that are working to make these networks of healthcare systems to start working. So I think it's more a question of our countries, our developed countries, to kind of enter with the technology, and then we just have to use the networks that already are set, and also to expand them to just make it more affordable and more reachable to everyone. And the thing is that these women want to take it. It's not something, you don't have the social problem of them not wanting to take them. It's just, you just have to make them reach these people. So, yeah.
Fran, do you have a question?
I do. It's quite prop related. So I really like the use of the plasticine and how you broke it down and then put it together and then swiped it across as sort of like a punctuation mark in the talk. Is that something that is used in your sort of field, or was that something that you came up with yourself?
So the bottom-up approach, I'm developing it myself. So it has like a lot of, this is a quite a theme that has been discussed in the industry, because no bottom-up approaches are not, so bottom-up approach is when you start from the molecular level and then you start to build up those particles. And the thing is that there are no current formulations that use this bottom-up approach, but actually the continuous process that we are developing it seems promising. So I think it can become a reality. So that's why this innovation would be so important. Not even not only for these specific kind of problem, like for the contraceptive usage, but also for the long-acting injectables in general that can treat a lot of diseases like chronic diseases, psychiatric disorders, and all of the diseases that require a daily pill. So that would be awesome. Yeah.
Thanks very much, Mariana. Well done. We're on to Tammy Strickland, who has a background in human health and disease, pediatrics, and regenerative medicine. She now works, though, in a research lab in RCSI that investigates the link between disrupted circadian rhythms, essentially your sleep-wake cycle, and various neurological conditions, including acquired epilepsy. And having previously taught biology and anatomy, Tammy really enjoys communicating science to anyone who's interested. So I hope there's no dozing at the back of the room, because school is in session. Judges, take notes. We will be tested on this afterwards. Tammy's talk is called The Curious Case of a Seizure at Night. Miss Strickland, the class is yours.
Picture this. I'm sitting on a certain fancy train from Paris to London. It's nighttime. I'm sitting in the dining carriage with a lot of other passengers. And then suddenly the lights start flickering. And then they go off. And then there's a thump and a muffled scream. When the lights come back on, there's a guy. He's lying on his back and he's writhing and his eyes are closed and he's shaking and people are screaming. What's happened? Who did this? And then the guy's partner calmly sits down next to him and she says to the crowd gathering, she says, he's having a seizure. He has epilepsy. And people look so frightened of this. And somebody says, does it run in the family? And she says, no, it's just him. And people look really confused and still look quite frightened. And then I'm sitting there quite confused because epilepsy is one of the most common neurological conditions in the world. It affects over 50 million people worldwide, including many historical figures. And a genetic basis of epilepsy is often not found. It only accounts for a very small proportion of people with epilepsy. So I go over to this lady and I sit next to her and I ask her quietly, did her partner take his anti-seizure medications today? And she says yes. But she says that his medications no longer really work very well. Nothing really works. And I'm sad because this means that he's part of the 30% of patients for whom no anti-seizure medications work anymore. So we lie him on his side and we wait for the 1 to 2 minutes for this event to dissipate. And while this is going on, a lot of the people in the crowd around us are talking about abnormal signaling and normal electricity and chemical imbalance and all these terms. And I stand up and I decide to clarify and I say, yes, a seizure involves an excessive electrical discharge from the electrical cells in the brain called neurons, and they can happen to anyone. But epilepsy is something different. It involves multiple seizures. It involves brain tissue and brain cells, electrical and non-electrical, being changed over time so that a person is predisposed to getting seizures. There's multiple culprits. It's complex. And somebody stands up then and says, oh, well, what triggered this guy to have a seizure? Was he photosensitive with all that flickering light? And his partner says, no, he has seizures at this time every day. And I think, curious, because you see, 200 years of medical literature has reported a similar finding, wherein people with epilepsy experience their symptoms at certain times of the day, and we don't understand why this happens. And exploring this in more detail may lead to the development of new treatments for people like this guy. It's just a matter of time.
I really enjoyed that, Tammy, and I have to say it was quite the transformation from the masterclass. So, folks, mystery on a train. Ruth, do you have any questions for Tammy?
I do. Well, I suppose I was just going to ask you sort of what inspired you to tell that story in that way, because it was great.
Oh, thank you so much. The reason I told that story in that particular way, because there's a lot of questions around whether Agatha Christie, a historical writer who wrote Murder on the Orient Express, she might have had epilepsy. There's arguments as to whether or not she had it, but a lot of people do support the notion that she had epilepsy. So she kind of inspired me a little bit. And the other thing that kind of inspired me was the sort of whodunit aspect. Because often in epilepsy, we don't actually know what the cause is. So that kind of whodunit aspect inspired me as well.
Thank you.
Tomas.
Tammy, can I ask you, why do you think we have such you're describing very well the reactions of the people around this poor man. Why do you think we have such a scared reaction when we see somebody having a seizure, when at the end of the day it's something relatively normal?
Relatively common, absolutely. That's a great question. So epilepsy has been reported for thousands and thousands of years. In fact, I think the first report of it was in 1000 BCE or something. It's crazy how often it's been reported. And I think historically it's been associated with things like madness, because there's a lack of control over the situation, and people can stigmatize behavioral traits like that, unfortunately, in society. It's a similar story for things like schizophrenia. I think people get quite afraid because it's the lack of control. It's a dramatic situation. It's something where people feel they can't help. I think there is certain, as I say, stigmas that seem to stick. One that I highlighted in the video was about photosensitivity, which actually only affects 3% of epilepsy patients, and yet people have this image in their mind that people with flashing lights, that's all there is, that's what sets people off. So I think there's just a lack of awareness in the public about something that affects over 50 million people. And that's something that probably needs a little bit more attention.
Great, Tammy. Thank you very much. All right, folks. On to our fifth FameLabber. It is Walter Stanley. He's a senior lecturer in mechanical and aeronautical engineering at UL. He has, let's just say, over 25 years experience specifically in the area of stress analysis. He says stress is something his students are very familiar with. He also researches composite materials, manufacture, and mechanical testing. And of course, his other main passion is aeroplanes, not flying, but he really enjoys just the idea of flight. So will our stress analyst crumble under the pressure of FameLab, or will his composed material have such a high yield strength that it comes out flawless? A little bit clunky, I'll give it to you. I am sorry for cracking so many material analysis jokes. I'm just having a moment. That last one, I think maybe 3 of you would have gotten. Let's hear Walter's talk, shall we? It is through a chink too wide, there comes in no wonder. Please welcome FameLabber Walter Stanley.
Hello, good evening. Did you know in 2019, people did it 4.5 billion times. So far in 2021, people have done it 2.8 billion times. And what's that? Well, get on an aircraft and take a flight. And it all started in 1903 on the sand dunes of Kitty Hawk in North Carolina, when the Wright Flyer 1 took to the skies, and it flew for 37 meters and it lasted 12 seconds. And the first passenger and pilot was Orville Wright. And here is a scale model of that seminal aircraft. And since then, we can see a modern aircraft down here, and the design has changed quite a bit. And when I get on an aircraft, I sit down and I look around in amazement, and I think, why are things designed the way they are? And I look out the window and I say, why is it designed like that? Well, it all goes back to the early 50s when the de Havilland Comet took to the sky. It was the first jetliner, and it flew higher, faster, and more comfortable than its contemporaries. But after 2 years of service, it was taken out because of 2 catastrophic accidents. And engineers needed to figure out, well, why did this happen? And so they pressure tested the aircraft, and after about 1,000 cycles of flights, they found hairline cracks starting to propagate from the windows, and windows at the time were square in shape. So that mandated a completely change in design, and that's why since then aircraft windows are oval in shape and they are reinforced locally. So a new design philosophy was born, and that is fail safe, and if it's going to fail, it'll fail safely. But besides that, significant fatigue testing must be done on all aircraft before they can fly. And thinking, well, why do the cracks start to happen? It's because of aircraft pressure. At 10,000 meters, the air pressure outside is a lot lower than it is at ground level. But we could not breathe at 10,000 meters. You would die after 60 seconds. So air is bled into the fuselage to about 3/4 of what it is on the ground, so that we can breathe easily. But that extra air pressure makes the fuselage expand outwards, and so significant stresses can develop. And other things, if you look at a window, you can see that there's a hole in the window. Wow, God, there's a hole in the window. And that's because the window is made up of 3 layers of acrylic. And the middle layer has got a hole in it that will balance the air pressure between the cabin and the outside. So what I'm saying is that aircraft flying is very, very safe. It's at least 2,000 times safer than it is to drive. So I can't wait to get back into the air and enjoy the fun it brings. Bye.
Do you know, I've always wanted to know what that hole in the window is, Walter. Thank you very much for finally solving that for me. All right, Fran, have you any questions as an engineer? I'm sure you have some questions for our FameLab, our Walter.
Yeah, that was definitely up my alley of interest there, Walter. And it was interesting because when you were talking, I was like, oh, I'm going to ask about the hole in the window, and you'd already covered it. So my question to you would be, which is your favorite aeroplane?
Oh, that is a really good question. Historically, just because of its sheer grace and beauty, I think the de Havilland Mosquito, because it was made by furniture makers, and it was made out of plywood, and it was one of the fastest aircraft in World War II, and it was an original composite made out of wood. Thank you. It's a great question. I love that.
Ruth, have you a question for Walter?
Yeah, I don't know. I mean, this whole failing safe thing, as a nervous flyer, Walter, I'm not sure I'm fully reassured yet by your talk. So tell me about some of the ways that aircraft fail safely.
Yeah, that's a really good question. It's just that what it is certain parts are designed to be replaced after 30,000 cycles, irrespective, because they can't fail safely, like the landing gear. They have to be replaced. But if, for example, a little stringer on a wing, if that breaks, that can break safely because the rest of the structure can bear the load that was taking. So it's still fine to fly until it gets down on the ground and then can be fixed. And aircraft are perfectly safe to fly with loads of cracks in them. That, I always say, scientists find problems, engineers solve them. So engineers will gladly certify it to fly, even though it's cracked everywhere.
Very good, Walter. Ruth's face when you said that was it. Walter, thank you very much. Okay,
cheers,
our 6th FameLabber, Debishmita Dutta. She's a material science PhD student, and she's studying magnetic nanoparticles and believes creative distilled teaching can make science simpler, exciting, and more fun. Debishmita has a BTech in chemical engineering, which she says she mostly uses to make a subpar cup of tea. And she says she's a lab nerd by day and a crochet granny by night. So she has spent the last few weeks weaving this story for you. Let's hope it doesn't unravel on her. Please put your hands together for Debishmita.
This is a banana, and this is my energy bill. This is all I've had since the past 10 hours to both write and produce my talk, whereas this is what your computer needs to merely process it. This is 439.32 kilojoules of energy, whereas when you do the math on it, it comes out to be 2,160,000 kilojoules of energy for a mere 8-hour computing time. That is an insane disparity. Why is that so? That's because your computers are not built like human brains. They are built with a von Neumann structure, which means they have a processor that processes the data and a memory that stores the data. These two communicate with each other and form a computer. Well, that doesn't seem great because we are now working on artificial intelligence and machine learning, where we are teaching computers to learn like human brains. Essentially what we're doing is we are forcing our brain's functionality on a structure that doesn't look like our brain. We don't have a processor and a memory that transfers information between each other because that would be very inefficient. A lot of energy would be lost between the transfer. Well, that is exactly what happens because a study says that by the year 2040, we are going to need 10 to the power 27 joules of energy to even do the same amount of computing that we do today. Well, that's not good because that is literally bigger than the world's energy budget we are clearly going somewhere wrong with the one-diamond structure, and we need to replace it with something that's a bit more brain-like, don't you think? Well, how does a brain work? It has a bunch of neurons with a synapse in between. There's a flow of charge in the form of ions or charged atoms, and there's an electric signal that is caused because of it. Flow of charge, electricity. Electricity is flow of charge, and that is exactly how your computers work. Is your brain a computer? Well, that is exactly the epiphany that Professor Mead had back in 1980s when he proposed the idea of a neuromorphic computer. A neuromorphic computer is nothing but a computer that looks exactly like a human brain. It has artificial neurons and SNNs, which are spiking neural networks. These neural networks are built exactly like your brain, as I said, which means they are solving both of our issues. They can now solve machine learning problems very efficiently, and they are also energy efficient because a neuron only fires up when there's an electric field between it, whereas your one-diamond structure is always consuming energy every time it is on. I think we've solved something. So next time you have a banana, remember, you and your computer might be consuming the same amount of calories very soon.
Excellent, Devishmita. I have to say, folks, my favorite moment of the last 9 years was Devishmita enthusiastically, earnestly, and hopelessly trying to explain complex numbers to me over what should have been a very short one-to-one call. Ended up being quite some time until eventually she gave up. Devishmita, well done, great talk. Okay, over to our judges, and let's hear from Tomas.
So the question is, given that now where everything is getting robotified, will the robot eat a banana and do the same computing tasks as if it was plugged to the electricity?
Not quite, but it would take the same amount of calories or energy to do the same amount of task, or even more in the future. So that's great.
Ruth?
Well, I guess, yeah, my question is if we can get computers to work with this much lower energy, what kind of opportunities does that open up? Because presumably that means we can do more computing.
Yeah, of course. So our brain is incredibly efficient, which means if you do the math, it comes up to somewhere around 10 to the power 15 times more calculations than a computer is capable of, because we do image processing, we do sound processing, and we don't have structured data input like a computer does. We have unstructured random stimuli that, we can take in and then process that data. But all of this sophisticated, complex work that we do, if we made a computer do the same work, it would take an immense amount of energy. So as you asked, that was a really good question. So efficiency is a big problem. So when we go into the future, when we get a bit more efficient, we could be solving a lot of issues in biomedical science, in image processing, in literally everything, because computers are used in every field today, right? So in reducing their the power intake is going to make every field impacted in some way positively. So that is great. That is great.
Thank you.
Devishmita, I'm sorry I was such a dreadful math student, but thank you for trying so hard.
Great.
Devishmita, everyone. Our next speaker brings us into the second half of our finalists. 6 down, 6 to go. This is Kevin Mercurio, who is a first-year PhD student at Trinity College Dublin, researching the impact of microRNAs on the gut microbiome. He's an avid blogger, published fiction writer, host of the Metaphorogens podcast, and Kevin has performed several science communication pieces on national and regional platforms and volunteers for SciComm initiatives like Science Slam Canada and Trinity's University Times paper. I hope you have the stomach for our gut researchers talk, because it's all about fear. It's called The Hunt for the Ultimate Scare. Now, will our judges be spooked easily? Let's find out. Please welcome Kevin Mercurio.
With Halloween coming up, I'm here live in Dublin's most sinister house, the residence of the FameLab serial killer, most known for their method of torture, death by PowerPoint. I'm sitting here alone in this house, devoid of any light or people. Sure, I'm scared, but this isn't a horror movie, and any one of you in our community of science enthusiasts would be as well. But that got me thinking, what is the scariest thing, really? What prompts the most amount of fear in the most amount of people? Adults, kids, blind people, deaf people, even people without complete function of their brains. Is it spiders, skydiving, scientific conferences? To answer that, we need to know a little bit about what fear is and its physiological consequences. Whether it's an emotion or whether it's the anticipation and actions towards perceived threats, fear is an evolutionary trait selected for in our ancestors long ago. Those who are able to fear and fear the right things increase their chances of survival. Fear of pain, of isolation, of the unknown are what researchers call innate aversions, all connected to death. But many people perform actions that go against evading death. Death can't be the scariest thing. Now, you may have heard the cliche that fear is in the mind. Well, I suppose the brain. Any physiological fear responses are dictated by small regions in our brain called amygdalae. Close to the primitive brainstem and the hippocampus memory center, amygdalae are what process our emotions to what we see and hear and determine external threats. Hey, there's something in the corner there. Is someone in this house? Anyway, the fight or flight response is typically associated with fear. You begin to hyperventilate to take in more oxygen. Pupils dilate to take in more light. Goosebumps appear, raising non-existent hairs to make us look bigger. Though we shouldn't be discouraged by these horrific experiences, since research has shown more fearful experiences increase our fear tolerance. But what about people without functioning amygdalae? Surely they should be included in answering our question. Well, research on patients with genetic diseases destroying that part of the brain have expanded the concept of fear. The most famous case study in fear research is that of SM, who understands fear but doesn't experience it, except in one case. Voluntary breathing of concentrated carbon dioxide induced panic and anxiety. And what's weirder was that control participants with functioning amygdalae experienced less fear within the safety of the lab setting. Therefore, the scariest thing that affects everyone is any uncontrollable threat that leads to elevated levels of carbon dioxide in your blood. So, if you can, remember to breathe this Halloween. And most importantly,
excellent. Let's bring up Kevin then. Kevin, brilliant talk. Really enjoy the theatrics. Let's start with Ruth. We'll go to Tomas.
Are you a horror fan by any chance? What was the inspiration there?
Hi, Ruth. Thanks for the
Hi.
question. Yes, I am a huge horror fan. Actually, me and my brother watch horror movies all the time. My favorite one, by the way, is Hereditary, just so everyone is aware. But yes, I've always been interested in learning about fear. I actually am one of the weird people who like being scared. I know that there's different reactions to fear, so that got me interested in it, as well as Halloween coming up, as it is one of the Irish holidays or occasions that I just learned about coming here. So definitely a huge horror fan.
Thank you.
Tomas.
I mean, can I ask you something? Because I'm fascinated by your talk, and you're saying that much of the fear is related to the fear of death. But for most people who are religious, there's the belief of the life after death. So then why should we be scared about death in your research?
Yeah, that's good question. Good question. So in terms of just, the research of fear, a lot of these innate aversions, what they call, are somehow connected to death. Obviously, there are, just like you said, religious people who believe in an afterlife. Other examples are people who just go into danger without really truly thinking about death. I can just think of like firefighters, for example. So it's not necessarily a conscious fear of death, but these innate aversions are connected to death and our survival in general.
Very quickly, Fran, do you have a question for Kevin?
I do. So in terms of fear, when does it stop becoming just a physiological response and actually become like a conscious response in terms of age?
In terms of age, what do you mean exactly? Like when people...
So like in terms of a baby, like when do they actually start realizing that they're scared rather than it just being a response that they've got no control over?
Yeah, thanks, Fran. That's a good question. So there are some research on young infants, I would think around 6 months or even younger than 6 months, that don't actually have some sort of fear response to typical things that we would normally fear. So there's still a lot of research that needs to be done, but it is likely connected to the experiences that they have observing parents, for example, or other people in their close vicinity.
Thanks very much for that, Kevin. Thanks, Fran. As a presenter of a live technological show, my fear is always technical difficulty. All right, our next speaker is Rebetswe Zwane. Rebetswe is originally from South Africa. She came to Ireland to pursue her doctorate and is currently in her third year of the PhD in Dublin City University. Her project is focusing on the computational modeling of pharmaceuticals. Away from the computer, Rhea enjoys writing, the sea, and quality time with loved ones. But one of the things Rhea noticed when she came to Ireland was that people were very interested in her hair. So she's going to talk about hair for us this evening. And after that, the judges will comb through the science and we'll find out, will it be a brief brush with FameLab or will Rhea take the crown? Could she go out with a bang or bangs? Let's find out. Rhea, over to you.
As a material scientist, I spend my time studying and thinking about the properties of the material around us, and I also get to design new materials with interesting functionalities. But as an African girl in the Western world, I am also forced to think about something else. Hair. Because of, can I touch it? Or, your hair is so interesting. I'm forced to think about the significance of hair because although hair protects us from UV rays and extreme hot and cold temperatures, politically hair still means a lot. On one hand, it can say something about ethnic origin and age, but on the other hand, it informs our social experience. Although forensic scientists can say something about you by looking at your hair, such as what medicine you have taken, but ordinary people can look at length, texture, and color and somehow judge whether you're considered intelligent enough, professional, or even whether you're considered beautiful or not. But as a material scientist, I have the privilege to not only think about the socio-political impacts of hair from my personal experience, but what gives hair its texture, color, and shape as a material. Hair is a biomaterial that is mostly made up of the protein keratin, the same stuff in your nails and your pet's claws. This protein plays an important role in how hair feels like and its mechanical strength. All hair, whether straight, wavy, or kinky, has the same structure. The inner part of a single thread of hair is made up of a long keratin chain, making hair elastic and rigid. That also makes hair an incredibly strong material, such that an average head of hair is able to support about 100 people. So as it turns out, Rapunzel could really let her hair down to pull up the prince and her family and her friends all at once. Hair grows from a tiny hole called the hair follicle. It is the pigmentation or the lack of pigmentation in the follicle, the size and shape of the follicle that gives hair its color and the differences we see across ethnic groups. Straight hair, commonly of Asian descent, sprouts from a round-shaped follicle, compared to wavy and kinky hair, where the follicle is more curvy and oval in shape, making hair grow out in curls or tight coils. If we had to get in the mind and matter of nature's creative process of making hair, deciding on texture, color, and shape, we'd find that nature, like a material scientist, does not only design for specific functionality, but also for beauty. Yes, our perspective of beauty is based on our experience of color, symmetry, shapes, and patterns, or even what we grew up seeing. But if we looked long and hard enough, we'd see beauty in the differences. Nature leverages shape, color, and pattern to give me my cotton candy afro coils, so that as a material scientist, I do not only have to ask what makes my hair beautiful, but what makes it strong. And if I had to let my hair down like Rapunzel, how many little girls can I pull up to let them know that their hair is beautiful, just as the original material scientist nature intended?
Excellent. I really enjoyed that. And I learned quite a bit from it, Rhea. Let's hear from our judges. I saw Fran smiling along to that. Fran, do you have a question?
Yes, I do, Ria. Is there any sort of reason, evolutionary-wise, in terms of why people in different regions tend to have quite different hair, but the same within that region?
Yeah, so like I mentioned, it's all about the size and shape of the follicle. And I'm not sure why, for example, maybe people who grew up more in Africa would have a certain type of shape of follicle or size and shape. But the differences are across, it depends how the follicle is also situated underneath your scalp. So it could be parallel to the scalp and then makes your hair grow like that.
Thank you.
What about Tomas? Do you have a question for Rhea?
Yeah, absolutely. And thanks for explaining. Now I understand why there's keratin in the shampoo when they're trying to sell it to me. But unfortunately, I'm going bald. So does that mean the keratin is not making its impact anymore? Is that the verdict?
So there are ways of like why hair loss really happens, and that also goes with age and genetics. And so that's all I could give you of what hair where hair loss comes from. But then as a material scientist, if I had to think about maybe what makes hair weaker or fall off, that has a lot to do with maybe the keratin, the protein that is there, or yeah.
Brilliant, very good. She was asking about hair. Her talk was about hair, Tomas. It wasn't about not hair. Thanks very much for that, Rhea. All right, our next speaker is Fernando Diaz. He is a PhD student at Tyndall National Institute. He's originally from Spain, and he decided to come to Ireland to learn English and fell in love with our beautiful country. Right now, he is working on the development of an immunosensor for the detection of microorganisms in plants. And even though his talk hasn't even begun yet, Fernando says he already feels like a winner because he's getting to explain microbiology to new audiences. So please put your hands together for our researcher of small things. Please welcome Fernando Diaz and his talk, Scope, Spectacular Filters Only for Farmers.
If Irish and Spaniards have something in common, is the love for beer, Malaga, and of course, the potatoes. I can get a direct flight to enjoy the weather of Malaga, and we both have found the perfect combination of beers. But regarding to the potatoes, our Irish and Spanish farmers are a little bit struggling trying to find the love of their lives, the perfect potato. But with my work, they will be able to find them from the comfort of their couch without the need to get dirty from head to toes. But how? Using my own dating app, Scope. In general, when you download an application to flirt, you need to have a very open mind, because you don't know what you can find there, and most of the possible partners suffer a type of anxiety and they just send love to everyone. On the field, it happens something similar. Some farmers are more focused in the quantity than the quality, and in terms of production, that represents a huge risk. This is a Scope. At a first sight, it seems like any other dating app, showing information as is the case of the location of the potato around the field or the size of the leaves. When it comes to dating, what we hate the most is wasting our time when someone is lying, and some of the potatoes could be hiding a very toxic information. Let's check one of these. At a glance, our lovely golden wonder seems like a nice potato, but she could be infected with, for example, Phytophthora infestans, cause of potato famine in Ireland in 1845, or with potivirus, responsible of the main losses in agriculture in Ireland. If any farmer chooses any of these potatoes by mistake, they will end up in a very toxic relationship that could destroy all the production of the year. But with a Scope, the farmer can avoid the contact with those liars using the appropriate filters. These filters are small proteins called antibodies, and each one of them is designed to recognize a unique molecule present in the surface of each pathogen, and that is what we call an immunocomplex. Each one of these unique molecules is called antigen, and the presence of them can determine the number of variants that could appear, for example, in a virus. If we activate the filters of our device, the internal mechanism will be able to recognize all the potatoes that are infected and generate a signal after the formation of the immunocomplex. Once this signal appears, the farmer can remove these potatoes before the disease spreads and focus on those perfect partners to maybe have their baby potatoes. So if you are interested in funding your starchy other half, just download the app, because the PhD student's salary is not enough when you want to bring your date for an exorbitant dinner. Thank you very much.
Thanks very much for that. Very good, Fernando. I had a toxic relationship with potatoes in my student years in DCU, where the local chip around the road was my local source of nutrition. I'm all over the place, I think, with the judges. Let's start with Ruth, shall we? Ruth.
That was great. Thank you. I loved your little potato characters. Could you tell me, could this be used for other crops as well? Or are you just starting with potatoes and then you're going to take over the entire vegetable dating world?
I want to have all the vegetables in the world. Like, it's part of my project. At the moment, I'm working with potatoes and I'm trying to detect potivirus in this case. And I will try as well in the future, if I have time before finish the PhD, to work as well with fungi. So hopefully I will have time. I will have more than one virus. But yeah, actually this type of sensors can work with potatoes, tobacco, tomatoes. It depends. Like, usually they are the same family, but the only one thing that we need is the sensor. After that, after you get the deposition of those filters that I'm calling, that are the antibodies, you can just analyze any type of target as long as you have that specificity in your surface.
Okay, thank you.
Mas, very quickly.
Yeah, can this process that you're developing help us better understand maybe the nutritional aspects of which are more nutritional and less nutritional? Because that's one of the problems of eating too much potato, isn't it?
Yeah, like, I don't know if we will be able to arrive to that point, but for example, this type of sensors that we are developing, they can help us as well to find allergens in the food. So in that nutritional level, I'm not sure, because you know that there are macromolecules as well. And like, for example, with the antibody, we are trying to attract one simple molecule, but in the future, I think that maybe in 20 years, of course, will be a possibility to get that nutritional levels as well.
Excellent, Fernando. Thank you very much. Thanks, Tomas. All right, our next speaker is Rashida Abdul Wahab. She's originally from Malaysia. She graduated with a medical degree and master's from Trinity College Dublin before obtaining a PhD from UCD in medicine. Rashida specialized in pathology. She currently holds the diploma of the Royal College of Pathologists in the United Kingdom. Outside work, she enjoys endurance sports, anime, and manga. Do not call them comics and cartoons, or I will come for you. Rashida's work is in looking at biomarkers for food to investigate exactly what people have eaten. So she theoretically could tell if you ate the last Ben & Jerry's from the freezer. But tonight, her talk is called Don't Judge the Book by Its Cover, The Truth About Weight. Please welcome to the stage, Rashida Abdul Wahab.
Once upon a time, there lived a couple called Homer and Marge Simpson. Homer struggled to lose weight, as opposed to his wife, who struggled to gain weight. Both of them suffered social stigma due to their weights. But science can explain why this is not their fault, why people shouldn't judge the book by its cover. Your gut can talk to your brain, just like an aeroplane signaling a control tower. With more than 100 millions of nerves lining its surface, your gut is like a giant sensor to foods, like skin to touch and nose to smell. When you eat, as food makes its way through your gut, hormones are released along the way that reflect not only how many calories are in your meal, but also its nutrient content, like how much protein and fat are present. They also signal the part of your brain that controls the appetite. When the signal is high, you stop eating. But as time elapses, the signals fade and you'll be hungry again. So, how much and when you feel like eating depends on how your gut talks to your brain. In Homer's case, his signaling is weak. He must eat large amounts of food before he feels satisfied. The opposite happens with Marge. She feels full quickly and stops eating, no matter how much she tries to gain weight. By understanding the biology, we can rewire the signal from the gut to the brain. And this can help people with extreme over or underweight. In my research, I'm hoping to help patients with cancer who lose weight unintentionally, but at the same time, it can also help patients with obesity, because these two groups are in the opposite end of the gut-brain signal. Your gut is beyond food absorption and excretion. It's so important that your rear end is the first body part you grew, but that's another talk altogether. Body shaming and weight stigma are enormously unhealthy. Sometimes what's in front of you is more than you know, more than you see. The truth is, there are many factors that influence our weight, some which are outside our conscious control. Raising awareness about the complexity of this issue will help reduce the stigma surrounding weight. So, the next time you meet another Homer and Marge, don't judge the book by its cover.
Very good, Rashida. Very good. Okay, let's hear from Fran. Fran, do you have a question?
I do. Could you name some of these hormones? Because you were very, it was like, oh, there are these hormones because there's ghrelin, is there, and other ones. So just give us a bit of an insight into what these different hormones are and how many of them there might be.
So we do have, let me just try and simplify it. So in terms of hormones that increase your hunger, we have ghrelin. I sometimes tell my patients it's like gremlin for them to remember. So ghrelin actually is produced by the G cells in the stomach. And then we have the opposite, the hormones that increase your satiety or your fullness. They are such as oxyntomodulin, peptide YY, and also GLP-1. And they are all secreted by the mucosa in a small intestine from what we call an enteroendocrine cells, L cells to be short.
I hate to think what the complex explanation was. No, I'm only joking. Very good. Ruth, your question.
I have maybe a really random question, but I read somewhere recently about different kind of dog breeds and that they're quite good models for hunger and satiety. And because I think is it golden retrievers never stop eating. I mean, do you use any particular animal models in your work? And can we learn from how other species deal with hunger?
I think I've read a similar studies that you've mentioned, but for my studies, we involve only human population. Okay. Yes, the Labrador, I believe, is the dog breed that has, there's a pathway in the brain, which is called leptin melanocortin pathway. And these pathways are very important in the appetite regulation. And any dysregulation from this pathway, which involves leptin communicating to leptin receptor, and then talking to POMC, and then talking to MC4R receptor, any part of this pathway that is affected, would lead to obesity. And that was the case from what you read about the Labrador, where they have, I think, a problem with the MC4 receptor, I think.
Thank you.
Very good. Thanks very much for that, Rashida. All right. Our penultimate speaker is Kyra Manai Hamilton. She's originally from North Wales, where she completed a BSc in bioveterinary science at Harper Adams University. She worked as a ruminant research technician there before undertaking a PhD with UCD between Teagasc and Ag Research in New Zealand. Her research concentrates on parasite fitness, and she literally runs a Hunger Games for microorganisms. But Kyra also has something else she cares even more about, and that's raising awareness. Kyra has dyslexia, and as you'll see, she does just fine, but she's made it extra hard for herself tonight because her talk will rhyme. Please welcome Kyra Manai Hamilton with her talk, Superpowers in Disguise, Life Through Dyslexic Eyes.
Kyra is my name, and I'm here to proclaim I'm not really a poet, but I'll try all the same. This is about my dyslexia and the trouble it brings, what it's meant to my life, and a few other things. As a child, I struggled with words and with reading. Words made no sense. I couldn't decipher their meaning. Letters on paper, they would swirl, twirl, and mash. 1 second I thought I had it, and then my brain would crash. The Merchant of Venice was an almighty struggle. The words and their meaning were just such a muddle. From letter spaghetti to alphabet soup. Sometimes sentences just looked like gloop. It made me feel small and stupid and slow. Well, my classmates understood, but my brain just said no. You've got to try harder. I would quite often hear, as if trying harder would make things more clear. Dyslexia, a name for the trouble with my brain. But wait, there is a way I can work without strain. Colouring my world in a purple haze helps me see out of this mental maze. Though commonly thought of as just being lazy, us dyslexics can't help that our brains are a bit hazy, backwards and forwards and all wrapped together. Learning may indeed take forever. Yes, perhaps we face a challenge or few, like reading and listening, to name you just two. But a brain that works different doesn't stupid you make. With tenacity and flair, we can make the world quake. Yes, being dyslexic can be a pain. But other benefits we some of us gain, strengths and weaknesses, come to we few. A curse or a gift, well, that depends on your view. Picasso, Da Vinci, Edison, and more. Einstein and Hawking were no dunces for sure. Talents abound in some, to be fair, like Goldberg and Knightley, the Fresh Prince of Bel-Air. Dyslexic brains are just different, not slow. Some things are harder, but others less so. Creating and solving are sometimes a breeze. Thinking dimensionally, some do this with ease. We all have our quirks, like food for the soul, to help us adapt and see life as a whole. Like purple, the colour I see the world through. It helps me understand things, just like you do.
Well done, Kyra. Let's hear from our judges. Let's go to Fran.
Hi, Kyra. Really interesting because I'm a recently diagnosed dyslexic, actually, about 18 months ago. So in terms of the purple glasses, could you explain a bit about how they work for you and why on a neurological level, if we know?
Yeah, so that type of dyslexia is called visual dyslexia. There's multiple different types, but that type comes with a processing problem, just the same as the other types, but this one can be improved slightly by the help of visual training and also things like different colors and layovers, which is why I use the purple. It stops letters moving for me as much, because otherwise it literally looks like alphabet soup. It's a lot of letters moving around, and I used to think that was perfectly normal.
Gosh. Ruth?
Yeah, thank you, Car. I mean, yeah, I was fascinated by the purple glass as well. I was going to ask about that, and I suppose now we're all in front of screens so much. You know, does it make a difference whether you're in front of a screen or reading a book? And do you have to think about layering different filters on to protect your eyes, but also to get that benefit from the purple glasses?
Yeah, so it depends. I have to have my brightness on quite low because the screen brightness can affect the way I read as well, because it happens to be the contrast just
plays havoc with my brain. So for that, I tend to color all the pages of anything I'm working on in purple, much to the frustration of my supervisors, who I occasionally forget to change the color back to white, and they know they're getting something from me if it's bright purple. But when it comes to books and stuff, it's a lot easier. So I just have a piece of purple plastic that I put over it. So I happen to use the shiny side, but other people use the matte side. It just happens to be the way my brain likes it.
Fascinating. Thank you.
Cara, thank you very much for that. Excellent. I'm sorry, I preambled your brilliant poem with my rubbish one. Thanks very much. Right, folks, we are at our final FameLab speaker, and it is Adam Stapleton, who, while often found spinning on his head or climbing the biggest thing nearest him, he holds an honours degree in theoretical physics from UCD and is currently pursuing a PhD in machine learning. Adam's research looks at the applications of machine learning in climate science, from predicting things like air pollution to understanding how the planet breathes. And his talk tonight is called Want to Save a Rainforest? And he's gone out on a limb here to leave us with a good climate story tonight that actually involves blockchain. So 3 cheers for our final FameLabber. Couldn't resist a corny joke to finish, could I? Ladies and gentlemen, it is Adam Stapleton.
Close your eyes. As you look around you, you see a wall of green, plants and trees of every species, and in your ears you hear the buzz of insects, the chirp of birds, and the howl of monkeys. The rainforest is deafening. But as you listen, the chorus of life fades. It's replaced by an ominous crackling. A wall of fire races towards you. Hot smoke burns your lungs, and your instincts tell you to run, but you can't move. Your feet are fixed to the ground. The trees can't escape these fires. They look to the sky, helpless. And a satellite looks back. It's only really from space that we can see the lungs of our planet burning. The Amazon is home to 390 billion trees, storing 180 billion tons of carbon, home to 50% of the world's biodiversity, and 1/5 of the world's fresh water. But we're losing 3 football fields of our precious lungs every minute as fires started to clear the land for soy and beef farming, logging and mining. Devastate the land. But from space we can see islands of hope, the protected lands of the indigenous people of the Amazon, pristine green, and researchers have shown that they're more resilient to fires. So how could we use technology to help the people on the ground, like these indigenous communities, protect the land? But David Down, an incredible group of researchers behind Gain Forest, are seeking to do just that using artificial intelligence and blockchain smart contracts. The artificial intelligence oracle uses machine vision, like you would find in driverless cars, to detect individual trees instead of roads and people. A predictive algorithm then forecasts which areas of the forest are most at risk of deforestation and prioritizes them in the cryptocurrency market. This connects people who want to protect the Amazon with people on the ground that can actually protect it. The donors contribute Ethereum cryptocurrency. The artificial intelligence oracle then tracks if the land has been protected. The people on the ground put forward a stake to say that they are protecting this piece of land, and if the land is protected over 3 months, 6 months, a year, then the blockchain automatically transfers the funds to them for protecting the land. Anyone can contribute to this marketplace once it opens, so maybe you could buy an NFT, a unique digital token that says, I protected these trees. And maybe you could show this to your children and grandchildren and take them to see them yourself.
Brilliant, Adam. Echoes of the brilliant Lorax by Dr. Seuss there, and channeling the same sort of drama. Let's hear from Tomas.
That was brilliant. I really got into the image. So I guess the challenge for you is, how do we scale this up? How do we make this a global movement of change from something starting in the Amazon?
I think for me, observing a project like this, scalability is built into it. So the use of artificial intelligence here is to do precisely that, to make use of the global network of satellites that we now have to cover the vast area of the Amazon. And I think in terms of scaling that then across the world, the challenge is probably demonstration of its viability. Does it actually work? And I think once this project, it's progressing into its final stage now. So they're opening the marketplace next year. And I think if it shows that it works, then that's when the funding will start to happen. That's when donors that have the money to be able to put forward to protect the rainforest will actually say, this is working, I'm going to contribute to this.
It's a really interesting idea as well, because it removes the agent in the middle, which is often, development agencies. You actually pay, because it's blockchain, pay directly to the farmers.
It's really.
Do you have a question? Sorry, Adam, go ahead.
Transparency and trust is really the cornerstone of the idea of this here. It's removing the hidden fees from banks, the fact that small plot holders like Indigenous communities can't get funding from large banks. They don't have access, and people in the global south generally don't have access to financial instruments like these. So I think it's a really brilliant idea.
I robbed a judge's question, so I'll ask one more from Ruth. Do you have a question for Adam?
Exactly. It sounds like an amazing idea. This currency, the Ethereum, is that what you called it, Adam?
Yeah.
How would Indigenous people be able to use that?
It's a great question. So Ethereum is a currency like any other. If, for example, I gave €1,000 to a member of an Indigenous community, they would have to transfer that to Brazilian reais anyway. So the idea is that you would then buy your local currency using Ethereum anyway. There are marketplaces with which you can do that.
Okay.
Excellent.
Thank you.
Excellent question. Excellently answered. Thank you very much, Adam Stapleton. And would you believe that wraps up all 12 of our FameLabbers for this evening? So our judges now have just 15 minutes to decide who will become the FameLab Ireland winner and go through to the international finals. They will head off to a breakout room to do this. So good luck, judges. I know it's not going to be an easy decision for you. Ruth, before you go, any final words?
No, I just want to say a huge thank you to all of the FameLabbers. You were all fantastic. I learned lots. I was inspired to go and read more and do more. So well done, all of you.
All right. Thanks very much, Ruth. Off you go to the judges' rooms. Now we're going to give you a wrap-up of each of those 12 talks so that you can vote. If you want to vote, you just go to famelab2021.com, or you can use the QR code. Quite fancy this year. You can use the QR code that will be on your screen now. Be sure when you're voting to pick one talk only. The winner will be announced on Facebook and Twitter later on in the show. So here they are again, our 12 FameLab Ireland finalists for 2021.
I'll be honest, the first time I saw the test for COVID-19 infections, I was afraid.
Do you remember Goldilocks? She's the porridge lover that snuck into the house of the three bears and went into their kitchen to taste their bowls of porridge.
I'm a free woman. I can do whatever I want, whenever I want, or at least most of the times. Sadie is not lucky as I am.
I'm sitting on a certain fancy train from Paris to London. It's nighttime. I'm sitting in the dining carriage with a lot of other passengers.
People did it 4.5 billion times. So far in 2021, people have done it 2.8 billion times. And what's that? Well, get on an aircraft and take a flight.
This is a banana, and this is my energy bill. This is all I've had since the past 10 hours to both write and produce my talk.
With Halloween coming up, I'm here live in Dublin's most sinister house, the residence of the FameLab serial killer.
As a material scientist, I spend my time studying and thinking about the properties of the material around us, and I also get to design new materials with interesting functionalities.
If Iris and Spaniards have something in common, it's the love for beer, Malaga, and of course, the potatoes.
Once upon a time, there lived a couple called Homer and Marge Simpson. Homer struggled to lose weight.
Kyra is my name, and I'm here to proclaim I'm not really a poet, but I'll try all the same.
Close your eyes. As you look around you, you see a wall of green, plants and trees of every species, and in your ears, you hear the buzz of insects.
So there were our 12 finalists. Don't forget FameLab 2021 to pick your talk. We will announce the audience winner later on in the show. So we're going to take a look back now over the last 9 years at FameLab Ireland, because 2021, as we said, is sadly the last year of the program. So we're going to do things a bit differently. Normally we would have an entertainment act as our interval, but instead we're going to take an indulgent and little trip down memory lane with some of our alumni. Have your tissues at hand and enjoy.
Once upon a time, long, long ago and far, far away.
There was a poem, there was sock puppets, there was props.
1, 2, 3, yeah!
What one word would I use to describe FameLab? Liftoff.
Unexpected.
Fantastic.
Energizing.
Entertainment.
Opportunity.
The sun will be shining, the angels are singing, and you'll just feel so happy.
Science communication through FameLab got into my veins.
FameLab invigorated my love for what I do.
So I felt like I was able to grow myself. I was able to meet new people.
I remember talking about the importance of stories an awful lot, and that was both really fun and also really important.
Without FameLab, I don't know if I would have been ever as confident to put myself out there or be trained enough to do that.
I'm Liz McBain. I'm the head of programme for the British Council in Ireland. FameLab was the first Science for Science programme that I was involved with.
It's been described as pop idol for scientists. It's not quite as cruel.
I first came here back in 2012 in some aspect. So we approached Science Gallery and we brought, I think it was 10 FameLab alumni, as they were then, from all different parts of Europe over to the Science Gallery to have like a showcase event. That went really, really well. And off the back of that then we thought, oh, why don't we give this a go in Ireland?
Back in 2013, it was the first year of FameLab in Ireland, and I was a PhD student in University College Cork at the time, and I had started to dabble in science communication, but FameLab really presented a really, really great opportunity for me to try my hand at it at a national level, and then as it turned out at an international level as well.
Yeah, you and your talk, Fergus.
Oh yeah, there we go. Why did I not get a haircut ahead of that talk?
What is it like watching it now? Or
it's actually great looking back at it. To be honest, it was exactly what I needed at the time. It was a real tonic to the research that I was doing.
Oh, wow. There was actually nothing in that cup. I feel like once I got on stage, I was ready to go. I was so ready to go. It was just a really great experience. I really enjoyed it.
Oh, it's crazy how still I am. I was shaking every second before it. It's a lot of anxiety. But the second I stood up, it just fell. And I had a really, really lovely time actually getting to say my message. And I had like one joke and it landed. And it just gave me this huge high. I was like, yes!
The weird thing is that all of the words kind of come back to me. I think I'd practiced it so many times that even a good few years on, kind of the memory is still there.
It gives this feeling of wanting it again. Nowadays, we are almost slaves to presentations. The format of the 3 minutes without PowerPoint, without slides,
and with the props, it's something that actually calls for creativity.
The training in FameLab for the national final with Malcolm Love, that alone was an amazing experience. Learned so much, and I'm still friends with pretty much everyone from the class of 2019 that year as well.
Yeah, the alumni network is brilliant. So I got to go to Prague to present at a FameLab International Echoes event there, and I went to Bulgaria to present at their International Science Festival.
Fergus McAuliffe, he was our first finalist, and he won the Irish national final, and then he went on to Cheltenham Science Festivals and he won the international final, and he also won the audience vote and he won the alumni vote. He won absolutely everything. It's just a pity that was 9 years ago, and much like the Eurovision, we haven't won it since.
That night in Cheltenham was almost well, it was career changing. There's no two ways about it. Like I went from someone who's doing a PhD and may have gone on to do further research to completely changing tack.
And one day I got a message from one of the members of NEFET into my email account saying, "Hey, we like your videos. Would you like to come and help us communicate to young adults about COVID?" The only reason I would have ever gotten this opportunity is from the training and the support I've gotten from FameLab.
One member of the FameLab alumni network, we actually ended up going to an emo rock concert together.
So do I have to admit now that I went to one of these emo concerts? Oh my God, I'm gonna kill him.
I think it might have been 30 Seconds to Mars.
Fergus claimed he didn't remember them. Yeah, whatever, he had a t-shirt.
So I think there are lots of things to be proud of. In any given year, we've 25 different partners. We've involved, I think, about 1,000 scientists and researchers in the programme over the last 8 or 9 years, and we've achieved an awful lot.
Now I think, early career researchers consider science communication as a kind of a facet of the job that's important in a way that when I was starting my PhD I wouldn't necessarily have thought about, and I think FameLab has contributed to that.
The end of it does make me a little sad. It becomes not just I took part in a competition or an event, it becomes something that I'm now part of.
There's now a network of researchers who've taken part in this, who take part in other events like Pint of Science, or multitude of other events that are spin-offs from FameLab or that were existing before FameLab.
Because we've come through this science communication crash course, we are now involved in the next steps of science communication. I don't feel unsupported in any of my future endeavours within science communication.
I think science communication in Ireland is in a brilliant place.
I can only say after the whole experience that I am so grateful.
To our funders, all our partners, all our participants, alumni, a huge thank to all of you. It's been absolutely wonderful working with you. It's been an amazing journey. Thank you so much.
Thank you very much, FameLab, for a brilliant experience over all the years, and for everybody involved in organising it. It's been a real pleasure.
Thanks, FameLab.
Thank you, FameLab.
Thanks, FameLab.
Thanks, FameLab.
My eyes watering now. Did you get that?
It is a bit emotional saying goodbye to this competition. It's been part of my life for so long. So thanks again so much for involving me in the journey, British Council. All right, it is time to bring back our judges, Ruth, Tomas, and Fran. Welcome back, guys. So, Fran, how did you get on?
It was really interesting, and it was really interesting in terms of us as judges, because we were looking for quite similar things, and we were looking for strong narrative, a storyteller that could not only know their research, but could use that research to take us away, take us on a journey, and put their research in place in the society that we live and make it relevant.
Excellently done, because I was worried you'd give away too much there. That was excellently done. I'm not going to give anything away just yet. So, 12 FameLab finalists come from hundreds of those competing up and down the country, culminating in this evening, in this very moment. So, who will be the FameLab Ireland 2021 winner? We're about to find out. First, though, the audience prize. So you voted during the intermission for your favourite FameLabber, And the winner will now appear across Twitter and the Facebook live stream now. Right. Okay. Very interesting. That winner may or may not be our overall winner for tonight. Let's find out. In third place with a €250 voucher is Tembi Fashina. Well done, Tembi.
Thank you.
Congratulations. How do you feel?
I feel great. I'm very pleased. Thankfully, everybody's talk was so beautiful. And I'm also really thankful to the people that helped me out with mine. Thank you.
Well done, Tembi. Congratulations. All right. Second place in the FameLab final is Adam Stapleton. Well done, Adam. Adam, how do you feel? Second place. You're muted.
So sorry.
He can't speak speeches, folks. He just can't put it into words.
I can't put it into words. All the talks were so good. Thank you to the judges. Thanks to British Council and everyone that was involved. And well done to everyone.
Well done, Adam. Well done, Tembi. So it has all come down to this. The FameLab Ireland 2021 winner and the last winner of FameLab Ireland ever is Tammy Strickland. Congratulations, Tammy. She looks absolutely confused. Tammy, well done.
I'm so confused. Oh my God. Thank you so much. That's a complete shock. Oh my God.
I warned them to have a good speech ready in case they won. Is this the best you can do, Tammy?
Listen, yeah.
Listen, well done. It was a great story filled with a really interesting delivery and lots of interesting science along the way. Congratulations. You are our FameLab Ireland 2021 winner.
Wow. Oh my God. Oh my God.
Okay, we will leave Tammy to try and find her words and recover her emotions. Tammy, congratulations. Guys, in all seriousness, thank you so much for a brilliant evening's entertainment. Thank you to all of our contestants. You were fantastic, and good luck to each and every one of you as you pursue science and all of the enthusiasm you bring with it to whatever role it is. Thank you to our judges, of course, to Ruth, Tomas and Fran. Thank you to our funders, Science Foundation Ireland, to our partners, CPL and Henkel, the British Council, of course, who deliver this and have done for years, the Cheltenham Science Festival, who brought this sort of event to life, and to News Talk, our media partner, and to you, the audience, for supporting this event every year without fail. It's a shame that this is a virtual one, but we know you are there clapping and cheering, of course, at the screen on your own at home. We'd love you to complete the FameLab Ireland final short survey. The slide's going to appear now and give you the information to do it. Please do. It'll take 1 minute, and it will be the same site that we use for the audience vote, famelab2021.com. That's it for me. After 9 years, I have to say goodbye on a FameLab evening for the final time. Thank you so much for watching. Good night and good luck.